mother strand, one strand will become leading strand, the other one become the lagging strand. Okay, this is the final product. One mother DNA will become two identical daughter DNA. So this is the mother, the one mother DNA. These two are the two daughter. DNA. And I make a mistake from the last slide. I apologize, sorry about that. The leading strand and the lagging strand, they do not refer to the mother strand, or it does not refer to template strand. It refers to the complementary strand. So originally in the last slide, the mistake I make, I called this the leading strand. No, this is the leading strand. Oh yeah, it's very bad handwriting. And this is the lagging strand. So the leading strand and lagging strand, they are referring to the newly made, the green, newly made complementary strand. Okay, the green one. That's the mistake I make from the last slides. Okay, sorry about that. Okay, this is a much better picture than I drew. This is a much better picture than I drew from my previous slide. Okay, you can see that the leading strand is the newly made green complementary strand. The lagging strand is the one that causes fragment. The fragment of the complementary strand we call it Okazaki fragment. Okay, Okazaki fragment. So see if you can label 5' and 3' here. So remember this rule, the newly made complementary strand, they are always made from 5 to 3. 5 to 3. Since you see the arrow of the DNA polymerase, okay, DNA polymerase here, the arrow tells you the direction. This is a green arrow, tells you the direction of the complementary strand is made. That means this part is the this is the 5' end here. For the complementary strand, we call it leading strand, and this is the 3' end here. Okay. What about the mother strand? The mother strand, well, if this is the 5', of course this will be the 3' end for the mother strand, and this will be the 5' end of the mother strand. Oh wow. And then what about the other one, the lagging strand? The lagging strand, of course, this will be the 5' end here, and this will be the 3' end here. Again, the end of the arrow is the 3' end. Origin of the arrow is 5' end. So when you label all these three, it becomes clear. Well, at least in my opinion. Okay, you may notice something. What is this purple thingy? This purple thingy, we haven't talked about this. This purple thingy is called primer. primer. Primer is like the starting of the replication. So when DNA polymerase bind to the template strand, the mother template strand, it will first lay down the it need the primer there. When the primer is there, then the DNA polymerase can bind to the primer, and then you will start the replication. So in other words, this will be the 5' end for the mother template strand. This will be the 3' end for the mother template strand. And you can see that the DNA ligase is sealing up the Okazaki fragments. Okay? And helicase is here. Helicase move to this direction to ups. Helicase move to this direction to open up the fork, and DNA polymerase move to in the same direction as the helicase. Okay? Well, I hope you understand what I talk about here. If you don't understand what I talk about here, don't worry. We have the video. Okay, this is a very good video. You can read